A very good morning, class 11th. Continuing with the today again with the structure of nephron only. We will talk about now more details in the nephron structure, which are going to help us to understand absorption and filtration function of the kidney, right? So first, see, actually based on the location, nephrons, they are of two types, that where they are present, like in our case majority of nephrons, they are of cortical types. So cortical, there are kind of nephron. They are cortical nephron and juxtamedullary nephron, but majority of nephrons, they are cortical nephrons. In a cortical nephron, what happened is the loop of, only the difference is actually between their height of loop of Henle, कि लूप ऑफ हेनले एक में छोटी है और एक में काफ़ी बड़ी होती है. सो इन जो कॉर्टिकल नेफ्रॉन्स हैं, देयर लूप ऑफ हेनले इज़ टू शॉर्ट. इट एक्सटेंड्स, इट गोज वेरी लिटिल इन द medulla. So majority of nephrons in the kidney, they are of cortical types. Juxtaglomerular is the in which loop of Henle is very long and run deep into the medulla. Just look at this diagram, you will be more clear. As you can see, these are our cortical nephrons. So only very short portion of the this loop of Henle is in the medulla area, and if the nephron is juxtamedullary nephron, then the larger part of loop Henle is so long and it extends completely deep inside in the medulla, right? This is that is the difference between cortical and juxtamedullary nephron. That in juxtamedullary nephron, loop of Henle is very long and it goes deep into medulla. That is the difference into the on the basis of type of nephron. Now today we will talk about Malfigian body, which is also known as renal capsule in the renal corpuscle in complete detail. Yesterday I told you that glomerulus and Bowman's capsule, they together known as Malfigian body and renal corpuscle. So first, if you look at this, this is our cup shaped structure, which is known as our, this is Bowman's capsule, right? Now this Bowman's capsule, it has a you can say it has a layer. It has cells which is known as, they have actually many layers in present in this Bowman's capsule. So here the inner side, this layer towards inner side in the Bowman's capsule, there is a layer with the foot-like structure. This wall is known as that, which is made of podocytes. This wall, it is known as visceral wall. This wall of Bowman's capsule, which is made up of podocytes, cells which have foot-like structure. Because actually podocytes word क्यों? Because they look like pseudopodia, like the false feet. They have this kind of structure. Because of their structure, this wall is known, cells, they are known as podocytes. This wall, which is made up of podocytes, it is it is toward the inner side of your Bowman's capsule. This is known as our visceral wall. Clear? Now the outer is having our squamous epithelium, and this wall covering it is known as our parietal wall. So two types of wall we are discussing here. That is, one is visceral wall, which is made up of podocytes, and another is parietal wall, which is made up of our squamous epithelium. Till this point we are going to make our squamous epithelium, and then this is our podocyte layer. Now then there is, as you can see, that in between these podocytes there are the gaps, there are the openings, and these openings, I am marking it with black, these openings, they are known as slit pores. This visceral wall has openings or pores or perforation bol do. They are not attached. They actually, they play a very helpful role in filtration, right? Now this Bowman's capsule is enclosing bunch of capillaries. This wider capillary, this wider branch of artery which is coming in this cup shaped structure, that is our afferent arteriole, right? Afferent arteriole is entering in, and then it branches out into the bunch of capillaries. They are narrow, so blood is coming with from the wider, and suddenly जो आपका पैसेज था वो नैरो हो गया. सो नाउ ब्लड इज फेसिंग विद हाई प्रेशर, क्योंकि सडनली यहाँ पे इतना वाइडर पाथ था और वो बहुत जाके नैरो हो गया यहाँ पे कैपिलरीज में. सो इट विल फेस प्रेशर, देयर विल बी प्रेशर ऑफ ब्लड अगेन, देन इट ब्रांच आउट इनटू द then the blood is moved out through the efferent arteriole. ये जो this portion of capillaries, it is known as glomerulus. Approximately 50 capillaries it has, right? Now between this capillary and podocyte layer there is a present a membrane. ये black dots, are you able to see? This is known as our basement membrane. It is present between your podocytes and the capillaries, right? क्योंकि यहाँ से जो भी absorption होगा it going to filter and coming through the podocytes, because podocytes they are helping us in filtration. So now फिल्ट्रेशन मेम्ब्रेन को अगर मुझे अलग अलग से लिखना है आप समझाने के लिए, फर्स्ट लेयर इज द दिस इज मेड अप ऑफ पोडोसाइट्स विच हैज स्लिट पोर्स, देन देयर इज बेसमेंट मेम्ब्रेन, एंड देयर इज what is this this whole, because these are playing a role in filtration. So this filtration membrane contains your podocytes, basement membrane, and then this is our capillary portion, right? And these are capillaries which are made up of endothelial layer, and in between the capillaries there, this is known as, these gaps, it is known as fenestrae, right? It is known as fenestrae. Now this complete 3 layer, but our concentration is on these 3 layers of the filtration membrane, which contains your capillary, right? Then basement membrane and podocytes. So blood जो भी filtered out होगा, अब ये blood coming through the afferent arteriole, this blood is coming through afferent arteriole, it filtered out here in the glomerulus. जैसे ही वो filter out होगा, it reaches at the filtration membrane, and then it is passing on to lumen. This space of our Bowman's capsule, it is known as lumen. Then सारा filtration होने के बाद it passes on to lumen of our renal corpuscle. Afferent arteriole क्योंकि बहुत वाइडर होती है, सो इट विल पास ऑन द ब्लड टू नैरो कैपिलरीज. अब कोई भी चीज़ ये पूरा वाइडर था, सडनली आपका पाथ नैरो हो गया, इट कम्स विद द ग्रेट एंड मोर प्रेशर, सो प्रेशर इंक्रीजेज, सो द फिल्ट्रेशन इज नोन एज हाई प्रेशर फिल्ट्रेशन. सो दिस काइंड ऑफ फिल्ट्रेशन इज नोन एज हाई प्रेशर filtration, because हम kidney का function we are going to discuss. That is why each and every membrane needs to be known to you. Now there in this after this, now we are coming to the PCT and further parts. So here as you can see that this afferent arteriole, it is through this blood is coming in, afferent it is leaving the blood, right? Basement membrane and slit pores. Now this parietal wall of Bowman's capsule, now it further now extended out into the PCT, right? So this red portion which I am making now is, it is very closer to our Bowman's capsule, or you can say renal corpuscle. So this portion is known as our PCT. And now this PCT is made up of, let's make it with the green, right? This PCT is made up of these finger-like projection. It is made up of basically brush border epithelial cells. As you can see, their structure brush border. This layer is known as brush border epithelial cells, right? And like this has brush border. Actually, this brush border epithelial cells, they have microvilli on their surface. Microvilli means they are going to increase the surface area for the absorption. So because यहाँ पे क्या हो रहा है इस portion में filtration, and when now filtration part is coming to the lumen area and then it is moving to PCT, एंड PCT में हम क्या डिस्कस करेंगे, वी आर गोइंग टू से कि जो फिल्टर होके प्लाज्मा हमारे पास पहुंचा है, नाउ फ्रॉम दैट पार्ट ऑफ प्लाज्मा और ब्लड वी आर गोइंग टू एब्जॉर्ब द थिंग्स, राइट. ये जो यहाँ पे जितना भी हमारे पास लिक्विड पहुँचा है, वी हैव टू एब्जॉर्ब ग्लूकोज, अमाइनो एसिड्स फ्रॉम दिस लिक्विड, राइट, फ्रॉम दिस fluid. So this has now this absorption area of PCT. It is made up of two things are more important here: brush border epithelial cells, as well as these cells, these brush border cells, they are having mitochondria. They have mitochondria. Why they need mitochondria? Actually, the things is that glucose, amino acids, they are 100% absorbed by these PCT. Sodium and potassium ions, 70 to 75% they are reabsorbed. They are actively absorbed in this PCT area. When we use the word actively, it means we need energy. So it means in PCT what is happening? Active absorption of substances. So we need here mitochondria. Which kind of epithelial absorption के लिए हमारे पास कौन से epithelial cells है? Brush border epithelium cells which have microvilli. So microvilli यहाँ पे surface area को और बढ़ा देते हैं so that more and more things can be absorbed. Now कॉर्टेक्स रीजन की अगर हम बात करें तो कॉर्टेक्स में हमारा क्या क्या आता है? ग्लोमेरुलस, बोमेंस कैप्सूल एंड PCT. They are included in our cortex and the rest is now in the medulla. Medulla में हमारा ascending limb, right, descending limb, this one is descending, and the loop of Henle. Glucose हमेशा 100%, amino acids 100%, sodium potassium 70 to 75% are absorbed in our which loop I should write here? PCT, proximal convoluted tubule. Now this proximal convoluted tubule, we are not, now I am not marking each part here. Now it is now continuing into your descending limb, right? Descending limb is covered with, this limb is covered with squamous epithelium. Whenever you draw this, always draw it little thinner as compared to ascending limb, right? This is thinner as compared to ascending limb. Ascending limb is little wider. Now this wall is, this ascending limb wall is permeable to water. It means it allow water, but it is impermeable to sodium and chloride ions. So इसका मतलब यहाँ पे जो fluid रहेगा that will be less concentrated because water will be more here. Why I am saying these points? Because when we will talk about our urine formation, then these things I am going to ask you each and everything, that why that is why we the purpose to discuss them in detail, that they are going to help us in during the time of urine formation. Again, descending limb is lined with, covered with squamous epithelium. This wall of descending limb, it is permeable to water but impermeable to sodium and chloride ions. So इसका मतलब इसमें water ज़्यादा है as compared to ionic concentration, और ये thinner होती है as compared to ascending limb. Now move on to your ascending limb. As you can see, then this portion is forming a loop like structure. So this ascending limb is little wider and thicker as compared to descending limb. Now it is opposite to ascending limb, descending limb. Ascending limb is opposite to your descending limb. It is impermeable to water and it is permeable to sodium and chloride ions. So it means when urine will come at this point, it will be more and more concentrated. Why? Because this portion is ascending limb is permeable, so more ions will be there, permeable to sodium and potassium ions, right? The last one is now that ultimately there will be our DCT. So next one is our DCT, right? DCT is lined with our cuboidal epithelium. Our PCT was covered with our PCT, that PCT is covered with our which epithelium? Recall that PCT has very special role. It is covered with our brush border epithelium. Where is the descending limb? Descending limb is covered with squamous epithelium. Ascending limb with the cuboidal epithelium, and DCT is also lined with cuboidal epithelium, right? It is permeable to sodium and potassium ions, and finally opens into collecting duct, and collecting duct is lined with columnar epithelium. So each and every epithelium, they have their particular functional value. That is why they are present in these particular tubular portion, right? Now the next we have to discuss is this branched blood flow through these loop of Henle and this tubular portion. There are the capillaries which are moving around these structures. So now I am going to discuss about these capillaries in detail, right? That what is these capillaries are actually, which kind of capillaries they are. Now listen to me very carefully. Yesterday I told you that the renal artery which is coming in, it further branch out into the few smaller capillaries, then they are joined together with the that is with the your open into renal vein which is carrying your deoxygenated blood, right? Now there are the many capillaries. First set of capillaries, where did we observe the first set of capillaries, that this afferent arteriole, ये कहाँ क्या करती है, ये एंटर करती है, इसके थ्रू ब्लड एंटर करता है, ये पर्टिकुलर ब्रांचेस छोटी कैपिलरीज में टर्न अप हो जाती है, एंड दिस पोर्शन वी वर सेइंग दैट ग्लोमेरुलस, एंड दिस ग्लोमेरुलस हैज़ हाउ मेनी कैपिलरीज? 50 capillaries. So this is our first set of capillaries, and in this area, in this particular area, there is a very high pressure is observed in this capillary bed. Approximately, this whole capillary set, it is known as capillary बेड, और यहाँ पे जो प्रेशर ऑब्जर्व होता है दैट इज 60 mm ऑफ मर्करी हायर इन द नेफ्रॉन साइट, हायर pressure when your afferent arteriole it is coming in and then bunch out into number of capillaries. This is our first set of capillaries, high pressure capillary bed, right? Next there is, now from this efferent arteriole, it covers the further branch out and it covers our renal tubular portion. It covers our PCT first, right? It covers our PCT. So this is our second bed of capillaries, and this second capillary bed, it is made of, it is known as peritubular capillaries. This is totally mentioned in our NCRT, and we have to pay very much concentration on this because many questions they come from this part, that this efferent arterioles, they further branch out on PCT with the help of a bunch of capillaries, and this capillary bed or number of capillaries, they are known known as our peritubular capillaries. Now these capillaries, because यहाँ से pressure low हो गया, now these capillaries they are under at low pressure, so this is known as low pressure capillary bed. So यहाँ पे जो pressure was observed, that is 13 mm of mercury. Clear? Now again there is the pericortical, then these peritubular capillaries, again then they cover whole around, move around this, they form a loop like structure around the loop of Henle. They cover the whole loop of Henle with its ascending limb with the loops. Basically these branch out into around the loop of Henle, complete loop of Henle, and then join with the renal vein, right? So peritubular capillaries, they rejoin to form finally renal vein. That happens in cortical nephrons, because cortical nephrons में जो loop of Henle है वो बहुत short है. But we have another kind of nephron also, that is your juxtamedullary nephron. So उसमें क्या होता है, in the juxtamedullary nephron, these peritubular capillaries, they goes deeper into medulla and they completely surrounds our loop of Henle, right? And these are known as, these capillaries, those capillaries which surrounds our loop of Henle, they are known as vasa recta. So I told you here about three capillaries, right? Three capillaries कौन सी? One is your peritubular capillaries, second one is your this high capillary bed, that is high pressure capillary bed. हाई प्रेशर कैपिलरी बेड कहाँ पे ऑब्जर्व होगा? ग्लोमेरुलस. देन देयर इज द थर्ड वन इज अवर वासा रेक्टा, एंड फाइनली this joins with renal vein, right? It joins with the renal vein. So this vasa recta, it coils around the loop of Henle and join into the renal vein. So what is the conclusion? That two type of nephron, cortical and juxtamedullary nephron. In cortical nephron, peritubular capillaries immediately join the renal vein, because हमारा जो portion है loop of Henle, that is very short. But in juxtamedullary, because this loop of Henle is very long, so these peritubular, they further coil around the nephron and they form, that is known as now vasa recta. So peritubular plus vasa recta finally joins our renal vein. This is all about information we need to understand about the capillaries as well as wall of PCT, DCT and ascending limb, descending limb in complete detail. Now it will be very easy for us to understand our next part, that is urine formation, right? So I will meet you with the next lecture.